Good afternoon, good evening. My name is Gwen Grenier from the University of Regina, and I am the National Coordinator for the 2023 CAP Lecture Tour. So now it gives me great pleasure to introduce our host for today's CAP Lecture Tour talk, Prof. Kimberly Strong, Chair of Physics at the University of Toronto, who will introduce our speaker.
Welcome everyone to the second talk in the Canadian Association of Physicists Lecture Tour series for 2023. As Gwen said, I'm Kim Strong. I'm the chair of the physics department and very happy to be able to host this event today here at U of T. We welcome the audience that's participating both in person, great to see some people here in the room, as well as those who are joining virtually. We wish to acknowledge the land on which the University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. And today, this meeting place is still the home to many Indigenous people from across Turtle Island. We're very grateful to have the opportunity to work on this land. We're also happy to host events such as this on the land here with participants from many parts of Turtle Island. This lecture tour is sponsored by the CAP Foundation and the Canadian Association of Physicists, or CAP, which is a national network of about 1,700 physicists working at universities, but also in industry and companies and research institutions across Canada. The CAP works on behalf of Canadian physicists to support physics education and research, and it sponsors a number of physics activities, including this lecture tour, as well as the national research conferences and the annual CAP Congress. The CAP also delivers a number of programs for undergraduate and graduate students, including research conferences, as I've mentioned, physics scholarships and prizes. So make sure the students in the room and online that you check out some of the prizes that you might be eligible for. And it's also a student advisory council. If you're not already a member of a CAP, please consider joining. It's Canada's largest network of physicists and physics students, and it's free to undergraduates. And for the first year of membership for graduate students is also free. I've actually been a member since 1991, so 30 years and counting continuously. I'm very proud of that. I joined as a grad student. So for the undergraduates in the audience, you can join even earlier. So I really do encourage you to join. It's a nice community to be a part of. Lots of opportunities for students. So now it gives me great pleasure to introduce today's speaker, Dr. Rowan Thomson from Carleton University. Dr. Thomson's research focuses on the development and application of computational techniques and theoretical approaches to study the interactions of radiation with matter, with an emphasis on research questions relevant to radiation therapy treatments for cancer. Today, Dr. Thomson will present her talk entitled Curing Cancer with Physics, Multiscale Modeling from Patients to Cells. So welcome, Dr. Thomson.
Okay, thanks for the warm welcome. It's a great pleasure to be here and see three-dimensional people, as well as thanks to everyone who's joining in 2 dimensions. So my talk today is geared at undergrads. And the first thing I want you to know is this research is very collaborative. And that means a lot of it is done by my students, and that's undergrads, graduate students, postdocs, as well as collaborators across the country and around the world. So we have a lot of fun while we do our research. This is a photo of us skating on the Rideau Canal just before COVID happened, and then we couldn't meet up in person anymore. This is pre-COVID on campus, and this is a conference this last June where people were together again. So work done by students, presented by a former student, but now a prof in physics. So if we look at the statistics, the lifetime probability of developing cancer in Canada is considerable. 45% of males, 42% of females are currently projected to have a diagnosis of cancer in their life. And radiation medicine plays a really important role in these treatments. So for example, for diagnosis, mammography is used to diagnose breast cancer or to screen for breast cancer. Radiation therapy is very commonly used as a treatment. So often we'll say radiotherapy instead of radiation therapy, but this is used for about 50% of cancer patients, either alone or in conjunction with surgery and chemotherapy. So I am a physicist, and I do research related to radiation treatments for cancer, and I'm going to talk to you about those today. So the focus is on how can we advance radiation medicine using physics. I'm not assuming a whole lot of background, so I'm going to tell you a little bit about radiation therapy and tell you what a Monte Carlo simulation is. And then we're going to go across different length scales. I'm going to talk about a type of treatment called brachytherapy, and talk about what we're doing in studying patients right now, patient scale stuff. I'll then go down to shorter length scales and look at cells and ask questions like how can we model a patient, cells, and nanoparticles if they're involved in treatment. And then I'll talk on even shorter scales looking at electrons interacting with DNA and then finish up. So first off, let's talk about some background radiation therapy and Monte Carlo simulations. So you may or may not know, but physicists have a big role in radiation therapy. I know in Ottawa, city of about 1 million people, we have 20-25 PhD physicists who work at the Cancer Center, ensuring people get their radiation therapy treatments. And in these treatments, radiation, typically in the form of electrons or photons, is used to kill cancerous cells. So this radiation can come from an external source, like this linear accelerator. So typically you'd have the patient lying on this black surface or treatment couch, and this linac will rotate around the patient and deliver radiation to them. Sources can also be internal, and here's an example. You can see these are like little grains of rice. There's radioactive sources in there, and they're implanted in the patient, and I'll tell you more about those in a bit. So how does radiation therapy work? Well, radiation enters into the patient, it travels through them, and it interacts with electrons and atoms. Energy is deposited in this process. There are broken chemical bonds, and we hope that those lead to biological changes that will kill tumor cells, kill cancer. Here's where physicists come in. So radiation dose is the energy deposited in tissue by radiation. And the units will be familiar. 1 joule per kilogram is the gray, which is the unit of radiation dose. So a doctor will say, I'm giving you a prescription for 80 gray of radiation or 145, whatever the prescription is, but it's physicists' job to measure it, calculate it, and ensure that those patients get that treatment. So I'm involved in the dose calculation side of things. In principle, if you were to look at this from a pure physics perspective, you need to solve radiation transport equations, but that's usually too complex and time-consuming to do exactly. So it's not feasible for most everyday situations, or it's just impossible. So we use approximate methods. Some of those approximate methods are used in hospitals, and they're really fast, but they may be inaccurate, from a few percent inaccurate to 90% wrong. So that's where some of my research comes in. And one of the approximate methods we use is Monte Carlo simulations, so accurate dose calculations using Monte Carlo methods. So you might be saying, Monte Carlo? What's that? Isn't that a place? Isn't that a casino? So yes, you're right. It's a very famous casino on the Mediterranean. It's known for gambling. So traditionally, gambling was done with dice. So think about a die. You roll a die, you get a number between 1 and 6. You get a random number between 1 and 6. So dice are random number generators. Monte Carlo methods are a group of approaches used to solve all sorts of problems from economics, physics, biology, finance, and so on that use random numbers. And this is how the name comes. It comes from the gambling connection and the dice. So sometimes if you were all here in person, we'd actually do experiments with dice. But as we're people in 2 dimensions and 3D, we won't do that today. So Monte Carlo simulations for radiation transport. We know the probabilities of different interactions of radiation with matter. So that's where a lot of the physics comes in. Random numbers determine which interactions occur, how far particles travel before they interact. So we use random numbers for all sorts of things, not dice though, machine generated random numbers. We track particles as they travel through the material, and we calculate the dose by adding up the energy deposited in the tissue. So if you're in second year or beyond, you know what I mean when I say a photoelectric interaction or Compton scattering. So if we imagine we have a photon traveling along through the medium and it undergoes an interaction, so let's say photoelectric effect or Compton scatter, we could have an electron that's set in motion from that. There could be a scattered photon or a fluorescence photon, so a photon coming from the atom moving to a lower energy state. The electron could emit Bremsstrahlung. That electron slows through various interactions in the medium, And in our Monte Carlo simulations, we model all of this. We keep track of the different particles that are created, and we model this shower of particles going through the medium. Okay, so you've had a lightning fast introduction to radiation therapy and Monte Carlo simulations and radiation dose. Hopefully you know what I'm talking about when I say those things. Now let's start talking about patient stuff and a particular kind of treatment called brachytherapy. So brachytherapy is up close radiation therapy. It involves radiation that's placed next to or inside the tumor. So I'm giving an example here. These are some small radiation sources. They're about the size of a grain of rice. Typically, they have a titanium capsule on the outside, something that won't interact with the body. And there are radioactive components on the inside. These can be put in the patient and left there just for a treatment period and then removed, or they can be put there permanently. They undergo radioactive decay and they just stay in the patient forever. So this is an example. The middle picture is for an eye treatment, where this part right here, imagine like a gold coin or a coin that has these radioactive sources on it. It's implanted adjacent to a tumor in the eye, left in place for a time and then removed. And this is a CT image of a prostate cancer treatment. And you can see the little bright spots in here are brachytherapy seeds or sources that have been implanted into the prostate, and those will stay there forever. So the goal of these treatments is to deliver high doses to the tumor, and you're able to do that because the sources are in really close proximity to the tumor, but then minimize doses to normal healthy structures where you don't want there to be radiation exposure. Of course, evaluating radiation doses is really important for these treatments. So in cancer centers, in clinics, in hospitals, they evaluate doses in an approach that I'm going to call here, and it's widely called the TG43 approach. It's an approach that was developed by Task Group 43 of the American Association of Physicists in Medicine. And in this approach, they assume that the patient is just a big bucket of water, and they know given positions for the seeds in the patient, so here shown in red in this right-hand diagram. And so if they know the dose distribution for one seed in water, then for a given arrangement of the seeds, they sum all those dose distributions up. So basically they have pre-calculated dose distributions around one seed, and then they do a superposition of those for a particular treatment. So this approach is great in that it can be implemented readily around the world in the same way. They can have extremely consistent results. But the problem is patients are not infinite buckets of water, right? So it's inaccurate. The effects of non-water tissues, shielding, the sources being present is neglected. And these can result in errors, and I'll explain some of them to you in a little bit. So then we might say, well, if not TG43, then what are we going to do? Well, we're going to come back to this picture of the patient and say we want to actually do model-based dose calculations where we accurately model the patient. We don't pretend they're a bucket of water. We model the tissues. We model the sources. If there are applicators or other things in the geometry of treatment, we model those as well. And Monte Carlo simulations are one way to do that. And clinically adopting these algorithms has been recommended. So there's a lot of work going into doing the physics and then getting it into clinical practice. And I'm going to tell you about a bit of that. So my group, Carelton Lab for Radiotherapy Physics, has been really active in developing Monte Carlo codes, and in particular for brachytherapy. So recently we developed EGS_brachy, which we call eggs brachy. It's called EGS because it's based on the eggs NRC package, which is the gold standard worldwide for this type of radiation transport simulation. eggs NRC was developed in large part at the National Research Council of Canada, which happens to be in Ottawa too. So I have collaborators there as well. So this code's flexible, accurate, fast, and it's also freely available. You can download it from our GitHub page and use it tomorrow if you want, or tonight. So to preview some of the things that we do with it, I've shown here some images that we generate of the simulations. The crazy colors are just because we can color anything how we want in simulation in computer land. So up in the top left, there's a palladium-103 breast implant where the purple and blue, if you can see them, are gland and adipose tissues in the breast. There are small green sources, if you can see them, that have been implanted in there. Those are brachytherapy seeds. Next one over is an iodine 125 eye plaque treatment, where the eye is not shown very well, but you can see the applicator. So this green part is a gold alloy plaque. Imagine kind of like a coin, but curved, domed. And then inside it are small radioactive seeds. And this is temporarily implanted in the eye and then removed. On the lower left is a preview of an iodine 125 prostate implant, where in red it's the prostate and there are small green sources, which are the radiation seeds. And there also, we can model things like a miniature X-ray tube source. And that's shown here on the right, where we model electrons being accelerated through the miniature X-ray tube, hitting a target. And then if you can see it, which I just barely can, I don't know if you can see it on the screen or on Zoom, but there are photon tracks what's being emitted from that miniature X-ray tube. So we can model all sorts of things with this code. I said we do model-based dose calculations. I said we model patients, but I haven't told you yet how we develop models of the patients to do simulations. So to do this, we have different collaborations with researchers at cancer centers, physicists, radiation oncologists. We obtain images, computed tomography or CT images of the patient. And here's an example of one for a breast cancer treatment. You can see rib bones here, a lung, and this is the target region, it's the breast. From these images, we know seed locations. So they're actually these bright spots in the image. And we'll also have a set of organ contours. So a physician, a radiation oncologist will have drawn contours saying, this is the target, here's an organ at risk. So we have all that information, that's our patient data. Then the first thing we need to do is use these images and use some algorithms to correct artifacts, because the presence of the brachytherapy seeds causes streaking artifacts in the CT images. So we correct for those, and you can see that those bright spots have disappeared. Then we assign tissue cross sections, so this is physics stuff, to millimeter-sized volumes of interest. And there are a whole bunch of these together, and those make up our patient model. And we give those volumes the name voxels, because each pixel in the CT image corresponds to a volume in our patient model. So a voxel is a volumetric pixel. And so you can see in this bright model here, this is our target region. So there'll be a mixture of adipose and gland tissues there. You can see the lung, you can see the ribs, and all the different colors there represent different tissues where we've assigned cross sections and mass densities, so physics properties. We insert virtual models of seeds in the patient. There could be 30 seeds, there could be 100 seeds. It depends on the type of treatment. And we have detailed models for every one of those seeds. So in this case, this is the palladium 103 seed. This is a lead marker in the middle in red. And these two parts here are where there's active palladium. And it's all encased in a titanium capsule. And from that, we're able to do these Monte Carlo simulations with a realistic patient model and of the treatment. So I want to give you a snapshot of some results. I'm going to focus on the two main cancers for male and female in Canada, for females breast cancer and for males prostate cancer. So let's get started with breast cancer. So of course, there are different types of treatment for cancer, and there are different types of radiation therapy for cancer. One type of breast cancer radiation therapy is breast brachytherapy. This is a permanent breast seed implant, PBSI, and it's in a form of accelerated partial breast irradiation. And they use the word accelerated because one of the alternatives is that patients go into a cancer center every day for a month or so and receive an external beam treatment. In contrast, this is done in one day. They implant the sources in the patient and then the patient can go home. So it's called an outpatient procedure. So this treatment is used for a form of breast cancer called ductal carcinoma in situ. And it follows breast conserving surgery, so lumpectomy. So the patient undergoes surgery, heals from that, then goes in for a one-day procedure to implant these radiation sources in case there's any cancer left that was missed in the surgery. And to give you an idea of the energy, there are palladium 103 seeds. So those are the bright spots in this target, which is shown in blue. and they emit photons around 20 keV. Okay, so relatively low energies. And in this image, I'm showing the skin outlined in red, which is an organ at risk of complications from radiation. So let's look at some results. So we're comparing full Monte Carlo simulations with the current approach that's used in the cancer center. So remember, TG43, patient's a big bucket of water. And so this figure is a slice going through the patient where the color scale gives a ratio of doses for the full tissue model, full Monte Carlo, relative to TG43. So that's what's done in the cancer center. So if they were all in agreement, we would be at 1, which would be kind of a green color, where TG43 is underestimating, we're in red, and where it's overestimating, we're in green to blue. So this is the target region here. You can see it's more green and blue. So in the target, doses are overestimated with TG43, which is a worry because there's a possibility of cold spots or underdosing. So the radiation oncologists or the doctors think, if they don't know about the Monte Carlo simulations, that the tumor or potential tumor tissue is getting less dose than they expect. That's a concern. And this could be as much as 30% lower. The skin is an organ at risk of complications because it's so close to the tumor area. And doses are underestimated with TG43. Monte Carlo doses, so the more realistic doses, can be upwards of 50% higher. And initially, physicians saw complications that they didn't understand based on the doses. And it's because TG43 was underestimating the doses to the skin. So there are side effects. Now let's turn to prostate brachytherapy. So this image I showed you before, where there are brachytherapy seeds implanted in a patient. So prostate brachytherapy is commonly used for low or intermediate risk prostate cancer. It can also be a boost with an external beam, so linear accelerator treatment for high risk cancer. And the typical radionuclide used for this is iodine-125, which emits photons of about 30 keV average energy. So my group worked with researchers at Centre Hospitalier Universitaire de Québec and Université Laval, and we completed the first large-scale Monte Carlo study and also looked at radiobiological modeling and outcomes modeling, looking at patients treated over about 10 years. So again, I want to give you an example of results. So over on the left-hand side, we have a color wash. Again, it's a slice through the patient, showing a voxel by voxel % difference between Monte Carlo and TG43. So if they were exactly the same, it'd be sort of a yellowy green color. So I'll draw your attention right in the middle here is the target. So it's labeled there. And you can see this patient has a large calcification in the prostate region. The calcification is all red. It has a much higher dose in reality than in the TG43 calculations. We actually don't care about that because we don't care what the calcification gets. But what is of concern is that the region surrounding the calcification here, you can see it's in blue, is getting a much lower dose in the realistic Monte Carlo simulations than in the bucket of water current clinical approach of TG43. And so if we look at this histogram of doses, so on the right-hand side, it's fractional volume, essentially a count of voxels divided by the total number of voxels in the target relative to the dose. The blue line is this TG43 that's currently used in cancer centers. And the blue is the more accurate full tissue Monte Carlo. And you can see that this TG43 approach is overestimating the doses to the tumor compared to the more realistic full tissue Monte Carlo simulation. And a big concern is these cold spots near the calcification that can be identified with Monte Carlo simulations, but not with TG43. So again, a concern about a cold spot, and we'll come back to that in a little bit. Now, only about 10 or 11% of patients have calcifications. Those that don't, I'm showing you results for a more average patient. So again, on the left-hand side, we've got a slice through a patient showing a voxel by voxel % difference. And you can see in the target region, there's much less variation in the doses between the Monte Carlo and TG43 approach. You can see there are small differences due to shadowing for the seeds. So ISA shadow, that stands for interseed attenuation. So the presence of the seeds attenuates radiation coming from another source. So there can be shadows. But then if you look at this histogram, you see that the blue curve for TG43 is only shifted a little bit upwards compared to Monte Carlo. So the Monte Carlo results are on average about 6% lower, which is considered clinically significant, though less of a concern than the calcified patient or patient with intraprostatic calcifications. So you might say, well, that's all well and good. Have fun doing physics, team, but does it matter? Does it matter for patient outcomes? So here's where we work with radiation oncologists and their teams, and they come in and do all the work looking at treatment outcomes. So we studied this and looked at 10-year biochemical relapse-free patient survival. And they found that it was 91.8% without intraprostatic calcification. So that's about 89% of patients. But for the 11% of patients that had intraprostatic calcifications, the survival was only 78.8%. So we found the treatment failure rate was higher for patients with intraprostatic calcifications than those without. And you remember those patients tended to have lower doses and cold spots. So this suggests that we need more accurate Monte Carlo dose calculations implemented in clinical practice. So we pitched that to the granting agency and got a grant to deploy eggs brachy across Canada. So the PIs on this grant, there's two physicists, Dr. Luc Beaulieu at Université Laval, there's me, and then there are two radiation oncologists, Dr. Juanita Crook, who's at BC Cancer Center and UBC Okanagan, and Dr. Eric Vigneault, who's at Laval and Centre Hospitalier Universitaire de Québec in Quebec City. And so we're working with partners at 5 cancer centers across the country to translate eggs brachy, this tool developed in my group, into clinical practice. We're using it to evaluate doses for patients already treated, and hopefully at some point use it to look at treatments that are happening. We're also doing something called radiobiological modeling, so calculating biologically effective doses, tumor control probability, normal tissue complication probability, and we're going to be looking at patient outcomes. So this gives you a glimpse of going all the way from physics to just developing code and using physics to applying it in real life and hopefully advancing radiation medicine. Okay, so down to shorter scales. Let's talk about cells. Let's talk about more multi-scale modeling and different ideas for nanoparticles in treatment. So far I haven't talked about cells apart from the outline, but cellular structures underlie the tissues that are modeled in the Monte Carlo simulations. Those cells are neglected for all the calculations I talked about so far, right? We're modeling these voxels in the virtual patient model. But the primary targets for radiation are cells and their components, which are much smaller length scale, right? So micron scale. So a big part of my research group and research activities of the last few years have been asking questions like, how can we do simulations from patient to cell, spanning all these different length scales? So there can be really long calculation times. There can be prohibitive memory requirements. We're talking core years for calculations to happen. So we have an allocation on Compute Canada, but even still, we need to be smart in how we do these calculations. So we're developing different techniques for this, and that's a lot of fun. But then there are also some great opportunities. So we're working with experimentalists to look at biological response to radiation. How can we develop experiments where we irradiate and then read out response in cells in a Petri dish, and also co-register with energy deposited to those cells? Now, I'm not going to talk about all those things today, but what I am going to talk about is developing new treatment approaches. So back almost 20 years ago now, people proposed, not me, to load up a tumor with nanoparticles, often gold nanoparticles, and then irradiate. And there were some important experiments done by Hainfield et al. In the early 2000s. What's the point of this? Well, if you load up the tumor with gold or some high atomic number element, you enhance photoelectric interactions in it because the cross section for photoelectric interactions scales with Z to some power with atomic number. So you can get more dose in the tumor, but then spare surrounding healthy tissues that can have less dose. And so there's a huge amount of research that's gone into this area by me, but also by lots of other people in the field. And physicists have often been working on how can we work out the dose enhancement factor that's achieved with gold nanoparticles. So this dose enhancement factor is just a ratio, dose to the tissue with the gold present relative to the dose with no gold nanoparticles present. So my group's had a lot of fun working on approaches to go from the tumor centimeter scale to embed a whole population of cells in that tumor. And then within each of those cells to model gold nanoparticles in different intracellular distributions. So we're developing approaches to model this physics across length scales and to calculate dose enhancements, often on the cell scale. So we calculate nucleus dose enhancement. So just looking at the nucleus of a cell. We calculate cytoplasm dose enhancement, just looking at the cytoplasm of the cell. And often we'll compare those to gold nanoparticles that are just spread out throughout the tumor, just to contrast what's happening in different modeling approaches. And we assess variation in these microscopic metrics or microscopic dose enhancement factors over tumor scale volumes, so over centimeter scale volumes. So I'm just going to give you a little snapshot of results. So what kind of gold nanoparticle distributions are we modeling in the cell? So here I'm imagining my nucleus and I'm modeling my nucleus as being a sphere, and it's surrounded by a cytoplasm compartment. So my cell is just a sphere, very simple. And the gold nanoparticles might be arranged around the nucleus, so perinuclearly. We're in a perinuclear organization. Sometimes gold nanoparticles are observed to be arranged in endosomes. So imagine, so in physicist language, I say these are blobs in the cytoplasm. And we can model different arrangements of these, but I'm going to show example results for 4 endosomes arranged in the cytoplasm. And we can also compare this to gold nanoparticles uniformly distributed in a tumor. So snapshot of results. These are results for 20 keV photons with a gold concentration of 20 milligrams of gold per gram of tissue. On the vertical axis, we're showing the nucleus dose enhancement factor versus depth or position in the tumor. Okay, so in centimeters, going from 0 to 3 centimeters. In red, it's this perinuclear configuration. In black, the dispersed, so gold nanoparticles uniformly distributed, and in blue, the endosomes. So you can see that these dose enhancement factors are varying in the nucleus. The dose enhancement factor to the nucleus is varying with position in the tumor and with the assumed gold configuration in the cell. Now you might ask what those shaded regions are. What we've done there is estimate the variation in dose enhancement factors due to variations in local gold concentration and in cell size, because not every cell is going to have the same amount of gold in it. They're not going to uptake the same number of gold nanoparticles. And there's also going to be a distribution of cell sizes in the tumor. So those are kind of like error bars. Now I'm showing the results next to it for cytoplasm dose enhancement factors for perinuclear, dispersed in black, and in blue, the 4 endosomes. And you can see here the difference in the scale, right? We're going from 4.5 down to 0 and 8 down to 0 for nucleus dose enhancement. So the dose enhancement factors for perinuclear are lower, which is because the gold is further away from a fair volume of scoring region. So a lot of the cytoplasm is further away. However, the dose enhancement factor is higher in the cytoplasm for the endosome configuration, at least near the front of the tumor, right? Because over here it's about 1.5, whereas here it's much higher. So this is an active area of research. We're continuing to calculate results, submit them to journals, and we have a paper on this under review right now. But hopefully that gives you a snapshot of how we can go from patients to cells to supporting the development of new treatment approaches. So now let's go to even shorter length scales and talk about electron transport, subcellular level. So low energy Monte Carlo simulations are of increasing importance. What do I mean by low energy? I mean, probably below 1 kilo electron volt, down to much lower than that. People want to look at these to understand radiation-induced damage on short length scales. So cell components, DNA, they want to understand the biological response to radiation and have tools to do that through simulations. So sometimes these types of calculations or this area of study is referred to as microdosimetry or even nanodosimetry because of the short length scales involved. So here's the problem. In most Monte Carlo simulations, the transport of electrons is treated semi-classically. So here I'm showing you a pictorial representation of an electron in a typical Monte Carlo simulation. So these blue X's are discrete interaction sites, and the cross sections for interactions may be derived by quantum theory. So if you're a higher undergrad physics student, you might look at this in 4th year physics, for example, maybe. And so this electron is assumed to undergo these free flight sequences where it has these discrete interaction sites. But many of you will know, 2nd year modern physics or above, that electrons are waves, right? They have a de Broglie wavelength associated with them. And so I'm showing the expression here for a particle of mass m. The wavelength is Planck's constant divided by the momentum. And if I assume a non-relativistic electron, using the non-relativistic kinetic energy relation to momentum, I can see that wavelength is inversely proportional to the square root of the kinetic energy. So as my kinetic energy decreases, my de Broglie wavelength becomes larger. So increasing wavelength with decreasing energy. So imagine you're someone who wants to model electron interactions with DNA. DNA is nanometer scale molecules, and my electron is getting a large wavelength. My approximation of the electrons being a point particle is probably not going to be a very good one anymore. So more than 10 years ago, with a colleague Ivan Kavrikov, we asked, at what energy does the classical Monte Carlo simulation approach of electron transport break down? And we actually used arguments based on Heisenberg's uncertainty principle, which many of you, again, will have seen in your undergrad classes. And we used general arguments and argued from this quantum theory side of things that Monte Carlo radiation medicine applications where electron kinetic energy is greater than 1 keV, and that's most of that patient scale stuff, that's fine. So the classical Monte Carlo approach is a fine approach in those contexts. But as the electron energy decreases below 1 keV, the validity of those classical Monte Carlo simulations starts to become questionable because this electron wavelength is becoming considerable. So more research is needed. And we've been working on that research over time, but it's very complicated. A full quantum model of radiation interactions in a patient is very complex to undertake. So we work with a simplified model. So one of the models we've looked at is a model where we have a small droplet of water. These red drops are point scatterers, which represent water molecules, off which electrons, which come in as plane waves, can scatter and undergo isotropic scattering. So this is scattering where it's the same in every direction, so S wave. We can fully model this and do calculations. I'll tell you a little bit more about that in a second. And we can compare this to classical trajectory Monte Carlo, where we have the electron going into the cylinder, undergoing these discrete interaction points and free flight segments, a lot like a ping pong ball, right? And coming out somewhere, and the water droplet is homogeneous. So we can do these two sets of calculations and compare them to try and understand where the classical trajectory Monte Carlo approach is breaking down. So let me tell you a little bit more about the quantum calculations. We're considering wavelengths between 0.03 nanometers and 0.62 nanometers, which correspond to energies between 3.9 EV and 1.5 keV. And of course, those are kinetic energies. We use random numbers to determine the point scatterer position. So those are these red dots, the water molecules. We constrain the average scatterer separation to be 0.311 nanometers. And then we vary the minimum separation between these scatterers to include structure. So water is quite a structured material. The molecules are not arbitrarily close to each other. So we assess how things change when we change the minimum separation. So in some simulations, we have Dmin equals 0.001 nanometers. So effectively the molecules can be almost right on top of each other, all the way up to 0.28 nanometers, which is more realistic of physical reality. So these quantum calculations involve numerically solving 1,000 coupled equations to find the electron wave field incident on each scatterer. For each set of model parameters, we simulate about 100,000 of these water droplets, each with different positions for these little red dots, so different scatterer positions. And then we average the quantum mechanical results over this ensemble of water droplets. And that's what we want to compare with our Monte Carlo results, our classical Monte Carlo results. So we can compute different quantities in each of the quantum and Monte Carlo frameworks. One of the quantities that we could compute, and I'm going to talk about today, is the droplet cross-section. And this is most easily understood in the classical Monte Carlo approach. So imagine you've got that cylinder and you divide it into polar angles. So where 0 is transmission and 180 is back reflection. And the number of electrons escaping the water droplet in the polar angle interval I divided by the number of incident electrons per unit area is the cross section. Okay, and so we can calculate a cross section for each of these polar angle intervals. We can then take those results and compute a relative error on the classical trajectory Monte Carlo results relative to the quantum mechanical. So the relative error is the sum of the absolute differences of the quantum and classical results divided by the total quantum cross-section. So again, I just want to give you a taste of some of the results. So here on the vertical axis, we have the ratio of the classical Monte Carlo to quantum cluster cross-section versus angle. So 0 is going straight through the small cylinder of water or water droplet. 90 degrees is coming out the side and 180 is going backwards. So these results are for per 15.6 electron volts, which is a wavelength of 0.311 nanometers. And there's no structure. So D minimum here is 0.001 nanometers and my relative error is 2%. So what happens when I introduce or I increase D minimum? So if I increase it to 0.1 nanometers, you can see I'm starting to get slightly bigger differences with this green line, and the relative error is 2.6%. If I increase D min to 0.2 nanometers, my relative error increases to 13%. And if I increase D minimum to 0.28, you can see I'm getting very large differences, particularly for forward scattering. So that's 90-degree angles or less. You can see there are big differences between the classical and Monte Carlo results. And the relative error is 21%. It's huge. Now let me show you a summary of relative errors versus energy. So on the vertical axis, percent relative error on the droplet cross-section versus energy. So these results are for elastic scattering only and no structure. You can see the relative error is less than 5% for energies above 15 eV. But let's see what happens as we increase D minimum. So as we increase it, you can see that the relative errors increase substantially. Again, I only have elastic scatter in this model. And now you can see the relative error is only under 5% for energies above a few hundred electron volts. I'm now adding in these broken lines, which are also adding in inelastic scattering into the model. The relative errors are increasing again, and the relative error is still only sub 5% for energies above a few hundred electron volts. And you might say, well, when do the quantum and classical results converge? And if we were to take 1% agreement as convergence, you can see that it's only for above 1 keV, which agrees with our simple arguments based on Heisenberg's uncertainty principle. But these were a lot more work to do than that. All right, I'm coming to the end and I think I'm in good time, so that's great. So I've given you a whirlwind tour of some of the research that I do. I've talked about everything from developing fast and accurate Monte Carlo dose evaluations using eggs brachytherapy, which you can all go home and download and use yourself if you want to. And I talked to you about how we're using that to do quantitative brachytherapy treatment assessments and improve treatments in partnership and in collaboration with doctors and clinical physicists across Canada. I talked to you about how we're developing new approaches for modeling radiation treatments across, or radiation transport across different scales from patients to cells, and looking at how we can support the development of new treatments involving gold nanoparticles. And finally, I told you about quantum effects in low energy electron transport and radiation interactions with DNA. I want to say one other thing before I go. So I'm a professor and Canada Research Chair, but I'm actually also Associate Dean, Equity, Diversity, and Inclusion in Science. And I come to this from lived experience. So various folks in physics in different groups are underrepresented, and they experience many challenges and barriers. So as students, you're the physicists of tomorrow, you're the physicists of today, you can affect change, we all can. And this is a selfie that I took with my 4th year undergrad class, 2019, back when all classes were in person. That was a 4th year class, thermo and stat phys. And we just had a lecture on partition function fun. But there were 25 students in that class and there were only 2 women. And that's only one aspect of diversity. So I think we can do better, but I encourage you all to be part of that change. And this is why I've done all sorts of things. I even drew my own cartoons for an article in Physics Today. So if you want to see an article, I think it's the first time ever there's been an elephant, a black hole, and a wrecking ball in the same article. You go look at that one. So I'll close by thanking the wonderful group of collaborators and students who make all this research happen, as well as all the institutional and funding bodies support. Thank you. Thanks for the invitation to speak.
So I'm the MC. So we're going to kind of alternate some questions that are being typed into this document here for me, but also questions from the floor. So why don't we start and see if anyone in the room has a question? Yes, go ahead.
Could you elaborate more on how the radiation distribution for brachytherapy is?
So, okay, so your question could have so many different dimensions to it. Do you mean how do we develop the model, or? So when we develop the simulation, we have a model of the patient, right? So we have all these different volumes of interest. Yeah, go ahead.
Specifically how radiation emanates from the seed.
Okay, so in each of those radiation sources, we've got a radioactive distribution in the seed. Okay, so you remember I pointed out the seed model. We know the distribution of radiation in the seed model, and we model photons initialized in the seed. We model them moving through that the radiation source, then they're emitted, so they can undergo Compton scattering, photoelectric interactions, anything that might be happening as they go through. Then when they get into the tissue, we model different interactions happening. So for the low energy, the 20 kV or 30 kV photons that are predominantly emitted by iodine-128 or palladium-103, it's mostly photoelectric interactions and Compton scattering. So things you'll learn about in 2nd year modern physics.
What information is encoded in a voxel?
So in a voxel, so you're talking about a volumetric pixel in the patient model. So we use the grayscale from those CT images. We map that to a mass density, okay, for the tissue in the patient. And then we will also say, aha, this is prostate material or adipose tissue or gland. And each one of those will have an elemental composition associated with it. So how much carbon, how much hydrogen, how much oxygen. We then know the interaction cross sections for those materials, and that's associated with each voxel.
Awesome. Thank you.
Thank you so much for your talk. I also wanted to ask you a little bit more about voxel-based analysis. I don't have any biology background, just a physics background. I wanted to ask you what's the difference between the voxel-based analysis and radiation versus the other methods that have been used so far?
So most of that stuff is on the physics side. So we're calculating, we're able to model the physics much more accurately in these voxels. So we can take into account the fact that in prostate tissue or in calcification in the prostate, different things are happening compared to water. Then we can use those results to do biological modeling, which I mentioned. I didn't talk about it in detail, but I said that we do it. So we've done things like calculate biologically effective dose, which is supposed to then be closer to biological response. You can then use that to calculate tumor control probability and normal tissue complication probability for these treatments. So the whole idea of modeling in these voxels is that we can develop these accurate models of patients, model the physics more accurately, that can then give us approaches to get to the biological response in a better way. Does that answer your question?
Yes, it does. Yeah.
Okay.
Thank you so much.
Thanks.
Thank you for the talk. So with this new method, which is different from whatever was used previously and better, but are there like any ways to experimentally record radiation dosage, like on some like fake tissue or something, to kind of test these models out? Or perhaps if you've tried to apply this method on some like actual cases and compared it to other methods?
Yeah, so they do, for radiation therapy treatments, there are different ways that they can measure doses. So sometimes they'll measure doses ahead of time in what's called a phantom. So they'll have like a water tank that they pretend is the patient, and then they can put a radiation dosimeter in it and do measurements. Then there are others who develop thermoluminescent dosimeters or other types of devices to measure radiation dose that's deposited during the treatment. It depends on the type of treatment though. So if you're doing an external beam treatment, say for lung cancer, you could put a detector on the patient's chest, and then you would measure the radiation on their chest, but the radiation would be delivered inside the patient. For some treatments, like the prostate cancer treatment, it would be very challenging to measure the radiation while the treatment's happening because all those sources are inside the tumor, and we don't want to implant some device to measure the radiation in the tumor because that's not good for the patient, right? But yes, there are approaches. So people will do experiments where they mimic the patient. So with different phantoms, could be a water tank, it could be something more sophisticated, or there can be different measurements for in vivo dosimetry, which is during the treatment. But that depends on the type of treatment.
And how does that compare to the results from, say, the TG43 model?
TG43. So it depends. is the big answer. Often we'll do well, I don't do them, but others will do measurements with water or a water tank. And there's excellent agreement if you're modeling what is being measured.
Yeah.
So we can do Monte Carlo simulations that model very well exactly what's being measured, and there's excellent agreement. So there's a lot of verification and validation that goes into these calculations, right?
Thank you.
Okay, so we have a question from our virtual audience. Given what you know about the shortcomings of TG43, do you have any thoughts on the ICRU's energy agnostic radiation weighting factors for converting measurements from grays to sieverts? For example, the weighting factor is 1 for X-rays, gamma rays, and beta rays, 2 for protons, and 20 for alpha particles.
Okay. Thank you. So probably not an undergrad asking that question. So this person is talking about maybe thinking about radiation protection, where we talk in terms of sieverts, but also thinking about, so different radiations will have different effects on tissues. They won't necessarily all have the same effect, okay? And so this is what the weighting factors take into account. No, I don't have a strong feeling about them. I stay out of that debate for now.
So with the kind of simple water simulation, TG43, the big issue was with the calcification in the prostate cancer. So have they tried putting, I don't know how the measurement goes for that kind of a model, but have they tried putting in something to block it, to create those shadows so they could measure it without the Monte Carlo simulation?
No, so they haven't been able to do that. So the interesting thing about the calcifications is, so the calcifications could be a sign of more aggressive disease, or it could be that the treatment failure rate's higher, or it could be that they're the cold spots. And I mean, we know there are the cold spots, but is that what's causing the treatment failure?
I just meant like, is it possible to just put something to block the radiation such that you see the shadows that you see in the Monte Carlo method? Because it seems like that's what the difference would be.
Oh, so you're saying do a TG43 calculation and then add something in to try and mock up the effect to get something like that? Yeah. What we're doing instead is saying, let's not try and fix the bad calculation method. Here's a better way to do it, and let's make that work. So we're saying this has been a great method for getting uniformity around the world, this TG43 approach, especially when they didn't have fast computers and enough memory. But now let's move to try and do more accurate calculations together. So that's what we're trying to do instead of fix the old algorithm. But what you're saying is, can we do correction factors to the old algorithm, right? That's going to be very hard. So people have thought about it.
I assume the model, it's hard to implement it into it.
Yeah, so you'd have to start adding different factors into that model, which gets really messy. So then it's sort of like, well, if you can do a more accurate calculation with better physics, just do that.
Always better.
Yes, it's always better.
I had a question about how easy it is to control the position in which of both where the brachytherapy seeds are placed with respect to the tumor, and when gold nanoparticles are placed inside cells. Like, can we control it voluntarily to make it become a 4 endosome or a perinuclear, or is it just we found that they settled in such a fashion?
So great questions, lots of them outside my expertise. So there's kind of two questions there. One is the brachytherapy seeds and the other is the gold nanoparticles, and they're actually a little bit different. So I have been in an operating room when they've implanted the seeds in a patient. That is done by a physician. So they stick almost like big needles into the patient, and then the radiation sources go through these big needles into the patient. So there's a lot of skill involved in that, and that's a radiation oncologist doctor that does that. The gold nanoparticles are delivered in different ways to the tumor. So one idea in the literature is that if you're doing it in a brachytherapy treatment, you have the brachytherapy seeds on strings, and the gold nanoparticles are on spacers in between seeds. And they're on something that dissolves in the tumor with time, and then the gold nanoparticles diffuse basically in the tumor. You can coat the gold nanoparticles with different substances, with different biological molecules or peptides. That's outside of my area of expertise, but it's a very active area of research investigating what different coatings will cause different uptakes in the cell. Can you target different parts of the cell? But again, that's outside my expertise, but it's a great question.
To my knowledge, there are like past, maybe not like directly for the medical field, but there have been like Monte Carlo simulations on like radiation and how like these like or like how ions interact with different materials. So like I know there's something called like SREM. I'm not sure if you've heard about it, but like, are there any major differences between like the Monte Carlo code that your group has developed compared to maybe like other attempts to use like Monte Carlo? Or is this like for this research area, it's like completely new?
So great question about different Monte Carlo algorithms. So I can say there are a number of different algorithms that are used for radiation transport. So the GEON4 is used a lot by the high energy physics community, especially particle physicists. It's also used in medical applications as well. It has different features, different benefits, different speeds for calculation, but it can model proton transport. EGS-NRC cannot model proton transport, but EGS-NRC is faster for photons and electrons, which is enough for certain applications. There's another code called MCNP, which is used for this type of simulation. The type of simulation you're doing, I'm not familiar, or that you're mentioning, I'm not familiar with, but there are going to be like thousands of different Monte Carlo algorithms because it's a technique that's used so widely. In this area, it's Monte Carlo simulations of radiation transport. And those can even have a lot of variation in them. But there'd be huge differences in Monte Carlo codes used for very different physics as well as finance compared to physics.
Thank you.
Thanks.
As I said before, I'm Gwen Grenier, the National Coordinator for the 2023 CAP Lecture Tour. And at this time, I would like to thank our Regional Coordinator, Professor Dan Zhao, the staff in the CAP office, the University Coordinator, and our host today, Prof. Kim Strong, for helping, thanks for helping organize today's talk. And thank you, Dr. Rowan Thomson, for such an amazing and excellent presentation. And thank you to everyone who participated. I also want to thank the CAP Foundation and its generous donors who have made the CAP Lecture Tour possible. Please consider making a charitable donation to support this program and many others. The third talk in the 2023 Lecture Tour will be given by Stephanie Chishak on Wednesday, February the 15th at 11 Eastern. And Stephanie will speak about neuromorphic hardware and its role in quantum technologies, and that will be broadcasted at Trent University live as well as online. And thank you to everyone again. Have an amazing evening. Thank you so much.